we're gonna do this like once every 2 years. Since you have something this year, probably gonna try to invite you back, maybe 2026.
Oh, that sounds great. Yeah,
Yeah.
thank you. Thank you.
You been to China before?
I have only been to China very briefly. So, and I would love to actually go and spend some time and meet
Yeah.
colleagues and travel. And there's, yeah, so many
Coast
great.
Harbor would be a good chance to look around. So we have this Cold Spring Harbor things like it's a series of conference like US. So it's just the Cold Spring Harbor Asia. They have a distributor in China. So they hold different topics. Actually, surprisingly, there's not so many immunologic topic, and this is probably for a very long time the immune tolerance theme. And then we want to have this as a tradition, like once every 2 years.
Oh, fantastic.
Yeah.
Hmm.
So um, shall we start it? I mean, I don't want to hold you too long.
Sure.
Okay. All right. Okay.
Uh,
Um, so let's get started. Hello everyone. Welcome to join our this today's Zoom seminar. So it's absolutely my pleasure to welcome our today's speaker, Dr. Shannon Turley from Genentech. Dr. Shannon Turley is currently Vice President, Senior Fellow in Immunology and Biomarker Discovery, Cancer Immunology Research Biology Division in Genentech. So a very, very brief introduction about Shannon. So Shannon completed her PhD training in cell biology in Yale, and following with postdoc training in immunology and immunogenetics in Joslin Diabetics Center in Harvard Medical School with Diane Christoph.
mhm,
So from 2004, Shannon started her independent research program and was appointed as assistant professor of pathology at Harvard Medical School and joined the Department of Cancer Immunology and Dana-Farber Cancer Institute. 2014, she transited to Genentech after she got her tenure at Harvard Medical School. So Shannon's research mainly focused on immunology and stromal biology. Her lab investigated the role of distinct stromal cell populations in mature and aging tissues, as well as their influence on shaping innate and adaptive immunity. Her lab is dedicated to elucidating the molecular interactions between lymphoid stroma determinants, myeloid cells, and lymphocytes, along with the functional implications of these interactions. The goal of Shannon's lab is to develop a comprehensive understanding of stroma cell function in inflammation and cancer, create innovative strategies to modulate the stroma microenvironment, and validate stroma targeted therapy aimed at enhancing protective immunity. Ultimately, her work seeks to advance the treatment of immunological disease and cancer. Personally, I really believe Shannon has made a significant contribution to fundamental concept in stroma biology, particularly in the context of the cancer biology. Her recent work on developing treatments aimed at restoring homeostatic fibroblasts highlights tremendous potential for improving patient survival and enhancing responses to immunotherapy. And in addition to her scientific achievement, Shannon plays a really critical role as a key member of the mentoring committee for the postdoc program at Genentech. Many of her trainees are now actively contributing to both academia and industry. It's really exciting to have you here today, Shannon. We are very much looking forward to your talk.
Oh, thank you very much for the really kind invitation and introduction. And I'm looking forward to sharing some of our findings with you and then getting feedback. So just so you guys know, this is work done by my laboratory at Genentech. Some of it started when I was at Dana-Farber, but it's mostly work done by folks in my lab at Genentech. I do oversee a therapeutic area where we're accountable for making drugs for patients with different immunological diseases. And we approach it by trying to target the immune system and also the tissue. And we take immunological and regenerative medicine approaches. So that's why I belong to this and lead this organization called Immunology and Regenerative Medicine. But I won't talk about our drug development work here. I'm just going to talk about more basic science. But I'm hoping you guys will see some of the themes in this work and how it might relate to what we do in drug development. And some of what I'm going to talk about today did inspire clinical trials, which I'm not going to talk about today, but we do have several therapeutics in the clinic that were inspired by this kind of work. And we're hoping that there will be more trials coming from this kind of work. So, all right, so let's see here. Okay, there we go. So many of you know, probably know that stromal cells play lots of different functions in tissues, and they function in healthy tissues and in diseased tissues. And interestingly, they also play, we think they play a role in a tissue's response to therapeutic intervention. My slides won't advance. Hold on. Oh, there we go. Okay, so in healthy tissues, stromal cells, first of all, can mean lots of different things. In this talk, I'm going to focus primarily on mesenchymal cells and for the most part on fibroblasts, although we do have some work on pericytes and endothelial cells, but I won't focus on those today. But in healthy tissues, fibroblasts play really important roles in establishing organ architecture. They deposit matrix and then remodel it to fit the needs of the organ. They also play important roles in establishing niches that support either the whole tissue or different cell populations. And we and others have studied these niches over the years. We continue to do some of this kind of work, and we have a couple of new stories coming out soon. I won't talk about them just yet, but looking at how fibroblasts support different immune cell types. So stromal cells can make growth factors for T cells, monocytes and macrophages, dendritic cells, neutrophils, B cells, but also endothelial cells and epithelial cells. And then they also, of course, make lots of chemokines and cytokines that promote either the recruitment and or the activation and differentiation of different immune cell types. They also play important roles in establishing lymphoid tissue compartmentalization and also how the lymph nodes can expand and contract during immune response. I won't talk about any of that today, but we also know that they contribute to wound healing. And when wound healing goes awry, they can become drivers of fibrosis. And actually this image here in the middle now is a lung from a patient with idiopathic pulmonary fibrosis, which I will come back to towards the end of the talk. The fibroblasts are the main drivers of the fibrosis in this diseased lung. And then we also know that they play roles in promoting tumor growth and metastasis. They can drive inflammation. They can underlie the formation of ectopic lymphoid tissues in addition to their role during development in establishing secondary lymphoid tissues. And then they can also contribute to vascular abnormalities. And so with all of these properties, we have come to understand over the years, we now at least can point to these cells contributing to diseases like rheumatoid arthritis, scleroderma, idiopathic pulmonary fibrosis, and other interstitial lung diseases, inflammatory bowel diseases, including ulcerative colitis and Crohn's disease. As I mentioned above, tumor growth and metastasis. We also think there's roles for fibrosis and fibroblasts in multiple sclerosis and other autoimmune diseases, diseases of the eye, such as age-related macular degeneration, and then, of course, different kinds of infections. And we know, for example, in long COVID, there's a presence of a very differentiated myofibroblast that seems to contribute to dysfunction in the lung. Okay. And then what's also super interesting from some of the work happening in the field and also here at Genentech, it seems that stromal cells surface in a number of diseases in patients who have different diseases, and particularly in patients who are not responding well to the drug. So we know that in inflammatory bowel disease, rheumatoid arthritis, cancer, and other diseases, patients that are being treated with immunotherapies, or also in the case of cancer, chemotherapies, there's an emergence of signatures associated with stromal cells in non-responders, which is super interesting for us. We're trying to understand how some of that works. And I'll talk a little bit about that in today's seminar. Now, there are some therapeutic options for modulating pathogenic fibroblasts in the clinic. There's only a few examples, and I would argue that the drugs that are out there are either not that good or have a very modest effect on patients, or they're really early in their clinical assessment, so we don't really know how they're going to work. But this is just a few of them on the right here in blue, and on the left are the diseases where these have been tested or are in clinical testing now. So like many of you, we think that understanding the complexity of tissues, both at the cellular and the molecular level, will provide key insights for making better therapeutics. These are images of that IPF lung. So this is a human fibrotic lung. And what you can see on the right are images of matrix that's been deposited. These are matrix fibers, collagen fibers. They're very densely packed and very aligned. And these are all being deposited by myofibroblasts. And it's like having a little trouble with advancing my slides. So, many of us studying human disease appreciate that we need to better understand what cells and cellular communities are present in these disease tissues. We need to understand where they're located and then what they're doing in disease, but also be able to relate back to healthy tissue to understand what they were doing there, and then understand the difference between health and disease. And then this can help us better understand what drivers of disease might be, and help us to develop better models of disease, and then also develop more accurate, effective, and also safe therapeutics. So this is really our central mission at this time is to really define the stromal compartment of disease tissues, and then figure out what parts of the stroma are driving disease, and then how we can drug it. So when I moved to Genentech, I was working initially in cancer immunology and immunotherapy, and I have since moved over to immunology. And we're working on many immunological diseases that are non-oncology related. But when I moved, I was working in oncology, and patients were being treated. This was early on in the immune checkpoint blockade revolution, and patients were being treated with drugs made by multiple companies, including this drug called atezolizumab or Tecentriq, which is a PD-L1 blocking antibody established by Roche. But responses to PD-L1 and PD-1 were very similar at that point, and we were seeing plots like the one on this graph. These are, sort of very classic spider plots showing what happens to patients' tumor burden after they're treated with an immune checkpoint blockade agent such as anti-PD-L1. So in green, you're looking at tumors, the tumor burden of patient after treatment with the drug, and in green, these are patients whose tumors are shrinking after being exposed to anti-PD-L1. So the tumor burden is being reduced, and you can see that in some patients it goes down completely, in other patients it goes down and then remains low over a very long period of time. So these are very durable responses. In blue, these are patients exhibiting stable disease. And then in red, these are patients whose tumors continue growing even after treatment with this therapeutic intervention, okay? So we have the responders, the stable disease, and then the patients who are progressing. And what we were trying to do was understand the mechanisms of response and resistance to clinically validated targets. And our goal then was to understand the mechanisms mostly of resistance, and with the hope that we could convert patients who were not responding well to the drug into responders. And then we could also, for the patients who were responding, would it be possible to increase the depth and duration of their response? So in many of the cancer trials that were happening with Roche drugs, we were in a very good position to often have biopsies of patients either before treatment. Sometimes we were lucky enough to have them before and after treatment. This is an example of biopsy data that we had prior to treatment. And then we're able to follow patient responses after treatment. And so this is just we had done some analysis of biopsies from patients who had advanced metastatic bladder cancer and had treated with atezolizumab, this anti-PD-L1 immune checkpoint drug. And we were interested in what were the molecular features and molecular pathways that were engaged in patients' tumors that were responding and those that were not responding, with a goal, again, of trying to understand what was going on in the tumors that were not responding well. So here in blue and in purple, these are molecular pathways that were enriched in tumors from patients who responded well to the drug. And down here in gold is one bar down here. These were molecular pathways that were enriched in patients who were not responding well to our drug. And what you can see is there was a lot of cytokine receptor interactions. And these were very interesting to us. And we began exploring all of these. And then we began really zeroing in on the TGF-beta pathway. So to sort of summarize this work, there was years of work, but I'm going to sort of tell you some of the main findings just in a couple of slides. So what we found was that patients who had tumors that looked like this, so this is called an immune-excluded tumor. This is a tumor from a patient, again, with advanced metastatic bladder cancer. And in these immune-excluded tumors, there are T cells, which are stained here in black, in the tumor microenvironment, but they have not actually penetrated the tumor islet itself. So this is a tumor islet, and here's another one up here. The T cells were on the premises, but they were in the tumor stroma out here in association with these collagen fibers, which were stained in purple. And you can see that there's lots of collagen in the stromal location where the T cells are. And these are highly aligned and densely packed collagen fibers. And if you look closely at the T cells, they have a polarized morphology suggestive of a cell that's, first of all, aligned with the collagen, and second of all, probably migrating on the collagen. And this tumor immune phenotype, which we call an immune-excluded tumor, represents about 50% of patients with bladder cancer and about 50% of patients across all human tumor types on average. And what we know about this tumor type is that it's less responsive to immunotherapeutic intervention compared with tumors that we call inflamed or hot, where there's lots of T cells that have made it into the tumor islet itself so that they're directly interacting with tumor cells. And then, of course, there's an immune desert or a super cold tumor, and those are tumors where there's no T cells at all in the tumor tissue, okay? So in these immune-excluded tumors, we found that expression in the tumor tissue of a TGF-beta response signature that was indicative of a fibroblast response to TGF-beta separated the responders from the non-responders. So you can see here in an immune-excluded tumor, the CRs, the complete responders and partial responders had a lower signal for this TGF-beta response signature compared to patients who had higher signal for this signature. These were the stable and progressive disease patients. Sorry, I had that. Yeah. So anyway, if you have a higher signal for the fibroblast TGF-beta response signature, you do worse on therapy compared to patients who have a lower signal for this signal. And this is a lower score for this signal. And this is only true for patients with an immune-excluded tumor type. So this one over here on the left. And then if you have a higher signal for this response signature, you exhibit reduced overall survival compared to patients who are de-enriched for the signal. So a lot of questions came out of these discoveries. And one that came out of it was, is there something about TGF-beta activated fibroblasts that was actively restraining T cell responses to cancer immunotherapy? Or is this just a biomarker of some other biology that's going on in these tumors and we needed to explore more? So we started asking whether we could combine a TGF-beta blocking agent with an anti-PD-1 or anti-PD-L1 blocking agent in immune-excluded tumors and potentiate the response to immune checkpoint blockade by blocking TGF-beta in combination. And we wanted to, of course, test this hypothesis in the appropriate tumor model. So we looked for tumor models in mice that recapitulated all of these features. So we needed an immune-excluded tumor that expressed where there was PD-L1 expressed in the tumor microenvironment, and that the tumor was only partially responsive to PD-1 or PD-L1 blockade. And we also had to have a tumor where there was active TGF-beta signaling and there was the fibroblast TGF-beta response signature was present. So at least we could say the stroma was responding the way it was in patients. So we found a few tumor models like that. And this is one example. So we treated the tumors in a therapeutic setting with either PD-L1 blockers or TGF-beta blockers or the combination. And what you can see, so the light blue is the controls. Peach here is anti-PD-L1 alone, and blue, darker blue is TGF-beta blockade alone. And this is an antibody that blocks all three isoforms of TGF-beta. And then in maroon, this is a combination of PD-L1 and TGF-beta blockade. And so what's striking in this tumor model is that PD-1 or PD-L1 blockade alone really has a very modest effect on tumor growth. So there's really only about 10% of tumors that are responding and regressing. TGF-beta blockade on its own had zero effect, but the combination led to about 70% regression in these tumors, and these responses were complete, so the tumors never came back. And this big regression that we saw was completely dependent on CD8 T cells, because if we depleted them, then we lost the response and tumors continued to grow. What was super striking was what happened to the location of the T cells. So at baseline, remember I told you that these were immune-excluded tumors, meaning there were T cells that already made their way into the tumor prior to treatment. And they were sitting out here in the stroma. So this is the tumor bed itself. This is the normal tissue. And there are plenty of T cells around, but they kind of sit at the boundary between the tumor and the normal tissue. And when we treat with anti-PD-L1 or anti-TGF-beta by themselves, we don't see any change in the location of the T cells. And this is enumerated here for total CD3 T cells and down here for CD8 T cells. But when we combine PD-L1 and TGF-beta blockade, things changed a lot. So then we started seeing an accumulation of T cells in the tumor bed itself. And these are pretty large tumors by this point. So this is a piece of the outer part of the tumor. But here you can see the normal tissue. So the boundary is here. And then if you, I'm not going to show it to you, but if you move into the center of the tumor, we would also see a pretty significant accumulation of T cells there. And also you can start to see necrotic tissue where the T cells are beginning to kill off tumor cells. So the preclinical therapeutic combination of PD-L1 and TGF-beta was reversing T cell exclusion, because we were getting T cells into the tumor and they were able to begin killing and causing the tumor to regress. So we wanted to understand better what was going on in the tumor. And what I'll tell you just very briefly is that there were lots of changes happening in the tumor microenvironment in this therapeutic combination. So we know that the stromal compartment, the T cell compartment, the myeloid compartment, and the tumor cell compartment are all changing in this setting. I won't go through all of that data with you today because it's just too much, but it is published. But what the two things that we found most striking was that the stromal compartment and the T cell were changes in the stromal compartment and the T cell compartment. So just, I won't go through all of this data, but we did do quite a bit of analysis on all the cellular compartments. And in the stromal compartment, we found there was remodeling of two fibroblast lineages or trajectories. And we found a very striking dampening of fibrogenic programs and also an augmentation of inflammatory programs in the combination setting. And so I think the best place to look at this is over here on the right in this heat map. So in these two columns on the left, these are changes in cancer-associated fibroblasts that we call TGF-beta activated or myofibroblastic caps. And on the right here in these three columns, these are data for inflammatory, or we call them IL-1 caps. And what you can see is that there was basically, in combination, we basically found a very significant change, sort of a shift from population 0 very big changes in population 0 and population 1. So down here in the combination setting. So we saw that there was a very striking upregulation of inflammatory genes in the fibroblast compartment and a downregulation of fibrogenic genes. So this is maybe not unexpected. You treat with a TGF-beta inhibitor in combination with anti-PD-L1, you lose the matrix programs and you begin to gain expression of inflammatory programs. So we found very significant upregulation of genes like associated with MHC1 and MHC class 2 presentation, as well as chemokines and cytokines. And then in the T cell compartment, we found something very striking, which was a pretty big progenitor exhausted population was shrinking. And the CD8 T stem cell-like memory cell population was increasing. So here we saw that the TPEX population was going down, but only in the combination. And here we found that the T stem cell-like memory cells were increasing, but again, only in the combination. And we validated this lots of different ways. And just to sort of summarize this part of the talk, we essentially discovered, and this was published a couple of years ago, that if you start with an immune-excluded tumor where there's T cells on the premises, but they haven't made it into the tumor islet itself, they sit out here in very close association with lots of cell types, including cancer-associated fibroblasts that have this myofibroblastic profile where they make a lot of matrix. And it looks like the T cells actually sit on the matrix and in close association with the fibroblasts, but they can't really make it into the tumor bed. But if we treat with PD-L1 and TGF-beta blocking antibodies, we see a very different picture. So now we have an inflamed tumor. The T cells have actually infiltrated the tumor very well, and they can now start killing the tumor cells. There is still some matrix out here and some immune cells and fibroblasts out in the stroma, but there's a very significant change to the fibrogenic programs out here, and you begin seeing less matrix and the fibers begin loosening up, and we think that that may contribute to the ability of the T cells to make their way into the tumor. And then this leads to a complete tumor regression, and we think just prior to the tumor regression, we get what we're calling a cross-compartmentalization, cross-compartmental licensing of the whole tumor environment by interferon gamma. And we found that every single cell compartment was showing a very strong interferon signature that was probably contributing to the tumor regression. Okay, so in parallel to that work, we were also trying to understand how stroma might be contributing to disease progression in pancreatic cancer. So pancreatic cancer is a highly aggressive treatment refractory disease. There are very few treatment options for these for patients with this disease. It's known that TGF-beta plays a major pro-cancer role in this disease. It's a very stroma-rich tumor, and many patients actually have an immune-excluded phenotype, which was surprising to us. We thought maybe all these tumors would be cold when we first started this study, but in fact, many patients had immune-excluded tumors. So they had the tumor type where there were plenty of T cells on the premises, but they were stuck out in the stroma, not able to infiltrate the tumor very well. So we wanted to understand using single-cell RNA sequencing technology. Again, this was published a few years ago, so it was still kind of a new approach, not so new anymore. Everybody's using it, but at the time we were trying to define the nature of stroma during tumor progression and pancreatic cancer. Using single-cell genomics, we used spatial mapping, lineage tracing, and then functional deconvolution. And we did this both in mouse and human to the best of our ability. We used a couple of different genetically engineered mouse models that had spontaneous tumor growth in the pancreas. And then we also made cell lines from these tumors and were able to implant them either into the skin or into the pancreas so we could have an easier system to begin studying the function of and the nature of the stroma in these tumors. So Claudia, a postdoc in the lab, took healthy pancreas and then pancreas that had spontaneously developed tumors at different time points, and then followed them, took tumors at early stages and late stages of tumor growth and profiled them lots of different ways. And this is the normal pancreas, and this is what's happening with the stroma distally from the tumors, and then this is what is happening in the stroma closer to the tumors. So you can basically see that throughout the pancreas, there's a very striking change in the stroma. This is just marked by a surface mark called photoplanin during tumor progression. And to make a long story short, we essentially found two major cancer-associated fibroblast subtypes in advanced cancers. So this is, I've sort of simplified this UMAP. It doesn't contain all the other cell types. It's only focused on the fibroblasts. And in greens here, this is what the fibroblasts look like in healthy pancreas. And then all the other subsets are fibroblasts that are found in pancreatic tumors that grow in the pancreas in these autochthonous models. And what we found actually, if you look at these blue arrows, we believe there were two main trajectories of fibroblasts from healthy tissue to early stage tumors to late stage tumors. And in these late stage tumors, again, we found these two main populations, this gold population, cluster 2, and this blue population, cluster 8. We tried to understand, we spent a lot of time trying to figure out how all of these clusters related to each other. And I'm not going through all the data with you, but essentially we believe that cluster 2 and cluster 8 were coming from we're evolving from through slightly different trajectories. And we were trying to understand what the drivers of these two clusters or trajectories might be. And we concluded from multiple approaches. So we did transcription factor binding enrichment and then gene set enrichment analysis. And we've taken some other approaches since then and concluded that there seemed to be one population, this gold population here, the bigger one, was driven by TGF-beta. And this smaller population, we thought was driven by some kind of NF-kappa B-like pro-inflammatory cytokine like IL-1 or TNF. Okay, so these were the two cytokine pathways that were inferred as being key drivers of these two major CAF subtypes in advanced mouse PDAC. And what we found actually is we studied these two populations very heavily and found essentially that these populations were changing a lot during tumor progression. And maybe you're not surprised, but I'll just show you a couple of examples. Oops. So this is what we were calling the TGF-beta CAF specific genes. And this row here, this is the healthy tissue fibroblast. This row here is the early stage tumor-derived CAFs. And then this row here, these are the TGF-beta CAFs in a late stage tumor, and their expression of all of these matrix-associated genes. So lots of collagens and related genes. So you can see that expression of these genes really comes up a lot in late stage tumors. And likewise, when we looked at those IL-1 activated CAFs or inflammatory CAFs, we saw that in late stage tumors, they express lots of chemokines and cytokines, suggesting they're probably interacting a lot with immune cells, maybe in different ways than the TGF-beta activated CAFs, but that they change enormously during tumor progression. We saw some changes that were induced in both CAF subtypes and also things that were lost with progression that are really interesting and published if you're interested in looking more at this. So then we went to the human tumors and we found a very similar picture. So we were lucky enough to be able to analyze normal adjacent tissue along with actual tumor tissue. So we did find a huge cluster of cells in the normal adjacent tissue. So these would be non-CAFs. And if you were taking pieces of tumor and you didn't have the normal adjacent tissue control, you might assume that these were CAFs, but we did know from the data that was available to us that these were from the normal adjacent tissue. So then in the tumor itself, we found these two main populations, 0 and 2. And this big pink one here, cluster 0, ended up being very, very similar to the TGF-beta activated CAF that we found in mice. And then this population was very, very similar to the IL-1 CAF or the inflammatory CAF that we and others, David Tufson's lab and others had described in mouse pancreatic tumor models. So single cell sequencing of human PDAC and normal adjacent tissue identified two major CAF clusters, including what looks like, again, a dominant TGF-beta activated subtype. And we were very lucky to find some markers of the TGF-beta activated CAF, particularly in late stage tumors. And we have not been able to find similar markers that really identify an inflammatory or an ICAF that makes them really distinct from all other fibroblast subtypes. But this leucine-rich repeat-containing protein, LRRC15, we found to be highly enriched in the stroma of mouse and human pancreatic cancers and also in other human cancers. And we made a gene signature for these cells, and we found that it was highly enriched in the stroma, but not in the tumor or in the epithelial compartment. And if you look here on the right, this is expression of just LRC15 itself. So this is a marker for these TGF-beta activated CAFs that comes up during tumor progression. We do not find it expressed in healthy tissues. You can see that it's up in many different human cancer types relative to the normal tissue counterpart for these tumors. So that we made the gene signature for both of these subtypes of CAFs, and we found that we were very happy to find that their expression pattern was quite conserved between the mouse and human tumors. So this is the IL-1 CAF signatures in human and in mouse. And likewise, this is the gene signature for the TGF-beta CAF. We generated it from the mouse data, but we found that it was very similarly expressed between mouse and human tumors. And then we made an antibody to LRC15 and tried to localize these cells in human PDAC on top and in mouse PDAC on the bottom. And what is super striking in this image on the top right is that first of all, there are a lot of LRC15 positive CAFs in these human PDAC tumors. And it's important to know that this is a transmembrane protein that's expressed almost exclusively on the membrane, the surface of these cancer-associated fibroblasts. So what you're looking at here is largely plasma membrane localized LRC15. So those are all TGF-beta activated CAFs. And what you can see here in the middle, these little islands, these are the tumor cells themselves. And so what you read in textbooks and you see in reviews all the time is that there's more stroma than there is tumor in a pancreatic tumor. And we really believed that after we saw this kind of staining. So there's this massive installation of TGF-beta activated CAFs marked by LRC15 relative to the actual tumor itself. And we found similar, but not exactly the same, but very similar patterns in these mouse to PDACs. So we were, of course, interested in the relationship between these cells and the immune cells, and then, of course, the relationship of these cells to response to immunotherapy. So we went to the human tumors and the mouse tumors and tried to co-localize, look for a possible co-localization of T cells with these stromal cells. And basically, in many of these human PDACs, we were observing a tumor immune phenotype of an immune-excluded tumor because we found CD8 T cells, but they were not really located in the tumor bed itself. And when they were in this peritumoral location, they were in very close association with cancer-associated fibroblasts marked by LRC15. So here you can see CD8 T cells. They seem to be sort of aligned with the swirls of matrix and the surface of the CAFs marked by LRC15. So about 60% of CD8 T cells were in direct contact or in very close association with LRC15 positive cells, both in the mouse tumors, but also in the human tumors. And what you can see here on the right in these mouse PDAC tumors is that the LRC15 positive CAFs, now marked in green in immunofluorescence images, are pretty abundant, and they can be found in very close contact with CD8 T cells, which are shown in white, but also with CD4 T cells, which are shown in red. And on occasion, you can find all three cells sitting together. So just to summarize this quickly, there were two major CAF subtypes that we found in PDAC. One that seemed to be activated by TGF-beta. We inferred this based on their gene expression pattern, so that we called this a myofibroblastic, or it was called referred to as a myofibroblastic CAF. And then another inflammatory-like fibroblast that seemed to be activated, we inferred was activated by a cytokine like IL-1 or TNF. We were, of course, interested in the TGF-beta CAFs because of our findings in the bladder cancer studies that patients who had a signature suggestive of a fibroblast activated by TGF-beta was associated with lack of response to cancer immunotherapy. We wanted to dig into these cells a little bit more, but we were interested to find that they were the dominant fibroblast population in human PDAC. And a signature score for these cells, I didn't show you this, but it's in our publication, was associated with resistance or lack of response to immunotherapy in multiple cancer types, mostly in those with an immune-excluded tumor immune phenotype. So the signature for the TGF-beta CAF was even better than the signature that I showed you in our Nature paper from 2018, where we found a signature for fibroblasts that were activated by TGF-beta in vitro and then applied to the human tumor gene expression data. This one performed even better in showing a separation between responders and non-responders to immunotherapy drugs. But I didn't show you the data, but it's in our paper. Okay, and then the TGF-beta CAFs co-localized in a peritumoral location with CD8 T cells, and we found a unique marker for this population, which is this leucine-rich repeat protein 15. Okay, so this led us to a couple of questions. So first was, what's the origin of LRC15 positive myofibroblastic or TGF-beta activated CAFs? So we spent a long time trying to figure this out, but during COVID, a couple of postdocs in the lab pulled together as much published an internal single-cell RNA sequencing data they could for fibroblasts from healthy tissues, both in mouse and human, and also for diseased tissue, both mouse and human. And what you're looking at right now is what we call the perturbed state fibroblast atlas. This is based on mouse data. Okay, so there's about 60,000 fibroblasts in this plot. And what you see grayed out here, these are fibroblast populations that you find in healthy and diseased organs, fibroblasts that you find in these organs. What was in bright colors here, these are the fibroblast populations that were emerging in specific organs such as pancreatic cancer, rheumatoid arthritis, and a couple of other diseases. And what was striking to us was that there are these LRC15 positive myofibroblasts, not only in PDAC, but also in arthritis and fibrosis and in different kinds of wounds. And based on this pseudotime analysis, we inferred that they were arising from these populations of fibroblasts marked by pi16 and col15A1. We also knew that these two populations both of them expressed a marker called dermatopontin, which will come into play in a few minutes. So we predicted, based on this kind of analysis and others, that the LRC15 positive myofibroblast was arising from healthy tissue fibroblasts marked by pi16 and dermatopontin, and possibly from this col15A1 relative of the pi16 population. And this was based on this in silico lineage tracing. So we love computational analysis, and we always try to follow up our predictions by using some sort of in vivo lineage tracing and or functional study whenever we can, though we can't always do that. So we developed a dermatopontin Cree ERT2 mouse. So these are fibroblast clusters from many different kinds of healthy mouse tissues. So now the UMAP looks very, very different. What we found in these healthy mouse tissues was that these same pi16 and col15A1 positive populations we found in the diseased tissues were also present in healthy tissues. And there they also expressed dermatopontin. So we made this dermatopontin Cree ERT2 mouse, and we were trying to understand then if we did a lineage tracing study with these DPT mice, could we generate LRC15 positive cells in a tumor setting that originated from the DPT positive cells? And that is, in fact, what we found. So we took the DPT mice and used them as a reporter, generated a reporter mouse, turned on the reporter, and then we implanted tumors and then tracked the cells over time. And what we found is that the majority of the LRC15 positive cells in these tumors were marked by YFP coming from the DPT positive cells in the healthy tissue fibroblast compartment, okay? So then we wanted to know what the molecular mechanism was driving these cells to emerge in tumors from these healthy tissue fibroblasts. And of course, a major and obvious question then was, is their presence driven by TGF-beta? So, and then what we also wanted to know was, is the development of these cells dependent on their capacity, the capacity of their healthy tissue counterpart to directly sense TGF-beta? So we use the dermatopontin Cree ERT2 mouse to ask this question. So do pi16 positive, dermatopontin positive fibroblasts that live in a healthy tissue, when they're exposed to TGF-beta, differentiate into a cancer-associated fibroblast that's marked by this leucine-rich repeat protein, LRC15? So we crossed the DPT Cree ERT2 mouse with the TGF-beta R2 flox mouse, so we could prevent those DPT positive healthy tissue fibroblasts from sensing TGF-beta in the tumor microenvironment. And so we turned on the Cree, we treated the animals with tamoxifen, turned on the Cree. We confirmed that TGF-beta receptor was knocked out in these fibroblasts. Then we implanted tumors, and then we waited a certain amount of time. This is just a summary of a bunch of mice showing that TGF-beta R2 was in fact very cleanly deleted in these cells. And in fact, taking this approach both in a subcutaneous implanted tumor model and also in an intrapancreatic orthotopically implanted tumor model, we found in fact that the LRC15 positive CAFs are greatly diminished when their healthy tissue counterpart can't see TGF-beta. So you can see here that the number of photoplanin-positive fibroblasts that express LRC15 is greatly diminished when those DPT-positive cells don't express TGF-beta2 in the tumor setting. And the data here, this is for the subcutaneously implanted tumors. And then this data here is for interpancreatically implanted tumors. The data is very, very similar. So this in vivo genetic approach sort of substantiated our in silico predictions, showing that these healthy tissue fibroblasts marked by dermatopontin give rise to the LRC15 positive cancer-associated fibroblasts in a TGF-beta dependent manner, where those fibroblasts directly sense TGF-beta during tumor progression and give rise to that calf population. Okay, so this is a mouse phenomenon. We wanted to know if it was also true in human. Not exactly an easy thing to address, but what we did was we took RNA sequencing data from 13 different human cancers, and we applied gene signatures that we made for the LRC15 positive cancer-associated fibroblasts, and also the cell that we think they originated from, so the DPT positive, Phi16 positive, healthy tissue fibroblasts. And we looked for enrichment of those gene signatures in these human cancers. And what was pretty striking to us is that when we found an enrichment for for the signature that's associated with the cancer-associated fibroblasts that's activated by TGF-beta, we found a de-enrichment of the genes that are expressed by the cell that we think they originate from, okay? And this was consistent with this data here on the right. So we found that the TGF-beta score in these patients' tumors corresponded really nicely with the expression of LRC15. And I'm not showing you the data here, but it also inversely correlated with the expression of these kinds of genes associated with the healthy tissue fibroblasts. Okay, so it seems like this potential developmental relationship between these two fibroblast subtypes, the healthy ones and the CAFs, was not only true in mouse, but also in these human tumors and across many different kinds of human cancer subtypes. All right, so then the big question was, do these TGF-beta activated CAFs, which seem to arise in lots of different cancer types, that seem to be associated with T cells that sit out in the stroma, so in this peritumoral location, associated with the tumor, the immune-excluded tumor immune phenotype, are they actively restraining T cell responses to cancer immunotherapy, or is this just true, and unrelated? The two coexist, but they're actually not really, the TGF-beta CAFs are actually not affecting the T cells directly. So we developed a system to address this question. Like any good immunology experiment, if you want to know the function of a cell type, the best way to test the hypothesis is to either take the cell type out or to put the cell type in and figure out what happens. So we developed a genetic tool that would enable us to selectively deplete those TGF-beta activated CAFs that were marked by LRC15 from established tumors, okay? So we made an LRC15 driven diphtheria toxin receptor mouse with a GFP tag so we could track the cells and enumerate the cells. So in this system then, we took tumors, or we grew tumors either subcutaneously or orthotopically, grew them to a significant size, and then we administered diphtheria toxin, right? So we waited for those CAFs to develop in those pancreatic tumors, then we ablated them, and then we said what happens to tumor growth, and then later I'll show you what happens to the immune response and what happens to the therapeutic response to checkpoint blockade. So first, when we got these mice, we were super happy to see how well they worked. So we were able to very extensively, and I'm not showing all the data, but also very selectively deplete LRC15 positive CAFs and not deplete other cell types directly. So here you can see the numbers go down pretty strikingly in this diphtheria, in this DT mediated system. And we were also really pleased to see that just depletion of these cells, so a single agent approach to depleting these CAFs, significantly slowed the growth of these pancreatic tumors. Again, true whether we grew them subcutaneously or we grew them orthotopically. And we actually saw some pretty interesting changes even in this single agent setting in the T cell compartment. So we saw a reduction in expression of some of these exhaustion markers, TIM3, LAG3, CD39, And then we also saw some gain of function in the T cells and their expression of TNF and interferon gamma. Okay, so significant delay in pancreatic tumor growth upon deletion of these LRC15 positive CAFs in a therapeutic setting. And we then wanted to know if this control of tumor growth in the single agent setting was dependent on the adaptive immune system or not. It could have also just been that these cells were directly impacting tumor growth. So it's known that CAFs can actually make a lot of growth factors for tumor cells. So it could have been that there was just a tumor cell CAF interaction, or there was a CAF T cell interaction that was regulating tumor growth. So Akshay depleted CD8 T cells in this system. So this is tumor growth in the control setting. And this is another experiment. So this is tumor growth upon depletion of LRC15 positive CAF. So again, as a single agent, you see a significant slowing of tumor growth. And when he depleted CD8 T cells, he lost the benefit of the depletion of the LRC15 positive CAF. So suggesting that tumor control in this setting is dependent on CD8 T cells. So then we wanted to know, is this effect due to some sort of direct interaction between the LRC15 positive CAFs with T cells, or is it because there's something changing in the tumor microenvironment as a result of taking those CAFs out? So maybe there's something coming up that's actually influencing the T cells. And the reason we ask this question is if you look over here on this little bubble plot, so we were monitoring what was happening in the tumor microenvironment upon depletion of those LRC15 positive CAFs over time. And this is what the naive skin fibroblast compartment looks like. And then this is what it looks like in a tumor at day 0. So you see these two main populations of CAFs. The red one is the LRC15 positive CAF population. And the blue ones are these other, they're not all CAFs, they're healthy tissue fibroblasts and CAFs. But there's these two main compartments. And what we found, if you look down here on the bottom, this is what looks like in a DTR system where we've administered DT. So these are all DTR positive. These are all DTR negative. All the animals have gotten DT. So you see the red ones go away. The blue ones remain because we're selectively depleting the LRC15 positive CAFs. And then this is at day 7, day 14. If you stop DT, what's really interesting is the LRC15 positive CAFs come back pretty fast. But we were interested at these two time points in the changes that were happening in the tissue microenvironment when we depleted the LRC15 positive CAF. So if you look at this bubble plot over here, you can see that in the DTR minus mice that have gotten DT, over time, there's an emergence of the LRC15 positive CAF. But there's a reduction, an overall reduction in the Pi16 positive progenitor fibroblasts that these cells come from, right? But upon depletion of LRC15 positive CAFs in the DTR positive mice, you can see that those cells go away. There's an emergence, there's an expansion, a relative expansion of the Pi16 positive cells. So we wanted to know, were the LRC15 positive CAFs negatively affecting CD8 T cells, or were the Pi16 positive cells having some kind of effect on the T cells? So we just took these two populations of cells out of the tumors and put them together with T cells in a simple activation assay and asked what was happening. And what we found actually, so this is the CD8 T cell compartment alone. So you can see that in black, those T cells are being activated. But if we put them together with LRC15 positive CAFs, we found that their activation was reduced. So it seems like the LRC15 positive CAFs can directly suppress T cell activation in this simple reductionist in vitro system. But if we put the T cells together with the LRC15 negative CAFs, we find actually that the T cell responses are not affected. And some of the folks in the lab actually think that these are significant, that there may be actually an immunosupportive function of those LRC15 negative CAFs. I'm not so sure about that, but at least they were not impairing the response. And then we also took, the folks in the lab took everything that was left over in the LRC15 positive depleted tumors and put them into culture with CD8 T cells, and again found that they were not restraining the CD8 T cell response. So this meant that the LRC15 positive CAFs do or can directly suppress CD8 T cell function, whereas LRC15 negative CAFs and any fibroblasts that are in that tissue are not doing that and may in fact even be immunosupportive. Okay, so then the big question was, if we deplete the LRC15 positive CAFs together with and administer anti-PD-L1, what happens? And you can see here that the combination was really significant in controlling tumor growth. So this is controlled tumor growth, control treated tumor growth, and this is tumor growth in tumors that were treated with anti-PD-L1 or treated with LRC15 depletion. So you see single agent activity in both of these settings, but the combination is really powerful in controlling tumor growth. So this suggests that depletion of LRC15 positive CAFs does in fact potentiate anti-tumor immunity in response to anti-PD-L1 checkpoint blockade. And this was also associated with really nice changes in T cell function. So we saw a greater proportion of granzyme B positive cells and also a greater proportion of these sort of polyfunctional cells with expression of interferon TNF and granzyme B. Okay, so I'm realizing that running out of time pretty quickly, but just to quickly summarize this last part, third part, there's one more small section, but basically we identified a peritumoral CAF marked by LRC15 that seems to be playing a special role in immune-excluded tumors. It evolves from a dermatopontin positive, Pi16 positive universal fibroblast, and it's driven by TGF-beta sensing by its fibroblastic progenitor. And it's a major subset in many solid tumors, mouse and human, and it seems to live proximally to CD8 T cells in these immune-excluded tumors. We think that it promotes some form of T cell exhaustion or dysfunction. We don't fully understand all of that yet. We're trying to figure it out. And that it seems to promote resistance to cancer immunotherapy. So this was a proof of concept that if we lose these cells, we can convert a non-responsive tumor in the setting of checkpoint blockade to a responsive tumor. And we can also convert an immune-excluded tumor into an inflamed tumor. So this was pretty exciting for us. We have been working with some of the computational biologists and scientists at Genentech trying to understand where else cells with these properties might exist. So we worked with a group that developed the searchable foundation model, where they were able to analyze publicly available single cell and single nuke RNA sequencing data across, it's now over 400 studies and across about 25 million cells. And then query this massive set of data for a score of a gene signature that we put into the system. And so we made a gene signature for human LRC15 positive cancer-associated fibroblasts. And what we found actually is that they're enriched in many human diseases. So we find them in systemic sclerosis. Here's the PDAX score. We find them in Crohn's disease, bladder cancer, of course, scleroderma. We find them in, this is scleroderma that manifests in the lung, and this is scleroderma in the skin. And then we find them in many different cancer types. We find them in idiopathic pulmonary fibrosis and in patient lung lung tissue from patients with COVID. Anyway, we find them in lots of different cancer types, but also other inflammatory and fibrotic diseases. And this is expression of some of the fibrosis-associated genes in the target organs of the diseases that were identified by this similarity search foundation model on the signature that we derive from PDAC CAFs, which you can see is present across all of these human diseases. And this is just a little bit of protein data confirming the presence of these cells that we've discovered in idiopathic pulmonary fibrosis, in scleroderma, inflammatory bowel disease. So this is fibrotic Crohn's. And then, of course, in different cancer types. So this is non-small cell lung cancer, and this is breast cancer. And I'm going to skip this slide for now and just go to the implications in future directions. So first of all, there are a lot of subtypes of stromal cells. Their states and their functions are complex. These cells change a lot during disease, and they also change during therapeutic intervention. We're interested in taking all of this information in because we're interested in defining markers, understanding the identities and the features of fibroblast and stromal subtypes that are associated with good properties in a tissue, but also pathogenic features. And we're hoping that we can determine the developmental paths, the location, the neighborhoods, and cellular communication associated with pathogenic and beneficial subsets. And then ultimately, can we elucidate the main functions of these pathogenic cells in different disease settings? Can we identify factors that drive their growth, their development, and their differentiation when they become pathogenic? And then ultimately, can this help us develop methods for controlling their function in different diseases? So can we either deplete them in a disease setting? Can we block their development? Or can we push them into another fate and hopefully bring about a better outcome for a disease tissue? So this is some of the folks that are in the lab. These are also, my lab is not this big. This is lab members and loved ones at a picnic we had out on Tomales Bay, north of San Francisco. But this was a collaboration with lots of people from Genentech, lots of clinical investigators who were kind enough to provide clinical samples to us. These are some of the past members of the lab. And thanks for your attention. I'm sorry this went a little bit longer than I had planned, but I'm really happy to take some questions.
Okay, Shannon, thank you so much. I think this is the very first time we actually have so comprehensive stroma biology. Okay, so we can start a question. Anyone questions can raise the hands or put your question in the chat box. Shall I?
Oh, hey Shannon, thank you so much for a truly inspiring talk. How much is the percentage of these LRRC15 positive cells in the total stroma population?
Yeah, so we really don't find them in healthy tissues. So that's the first thing. So if you look in most healthy tissues, you won't find them. Second, the RNA is really difficult to find in single cell. So just so you know, it's always very weak. It's always hard to find, but you can find it better in bulk seq, and you can really see it in either by flow cytometry or by IHC. So unfortunately, it's one of those genes that a lot of people miss because it's not easy to find. But when you get good at finding them, it depends on the disease. So if you look in human fibrosis or in human PDAC, amongst the fibroblasts, they're pretty abundant. But if you look in ulcerative colitis, they're very hard to find. If you look in Crohn's disease, if you have a patient sample that has like a fibrostenotic lesion, and you've been lucky enough to sample the lesion, you'll find them. But if you get a different part of the gut, you may not find them at all. So it's heterogeneous. It depends on what part of the tissue you're collecting, and it depends on what disease and stage of disease. So in rheumatoid arthritis, we haven't done this work, but others have certainly found them. But they live in certain parts of the tissue, and they're not, uniformly present. So it just depends on where you sample, when you sample, what disease.
I see.
Yeah.
So in the DTX model, you have about how much percentage of the fibroblasts are the LRRC15 positive?
Well, of the podoplanin positive, it depends on the stage of the tumor, but it can be up to 50%. But if you're in early stages, it's progressive. So go from 10 to 20 to 50. But it also depends on how you sample the tumor. So
I see.
Yeah.
Of course, I guess a related question is if we deplete similar